Today, we are honored to welcome with us Dr. Thomas Gurry, whose presentation is entitled Leveraging the Gut Microbiome Towards Chronic Disease Prevention. Dr. Gurry, the floor is yours.
Thank you very much, Dr. Katran, and it's a great pleasure to speak with you all today. I will be sharing some slides, so I'm going to share my screen if that's okay. But yeah, I look forward to discussing this all with you afterwards. I'm going to do basically a little bit of a survey over what some work that I've been working on and been involved with, in particular in the gut microbiome space, and specifically how it can be used to leverage essentially all of its capabilities towards disease prevention. So this is going to be a pretty narrow lens on this question that I'll be showing. It's a very large question. The microbiome is now a huge part of human health. You know, what used to be something that was considered, you know, kind of fringe research is now pretty much established as centerpiece in immunology and all sorts of other fields of medical science. So I by no means am going to present an exhaustive overview of this question of how the microbiome can be brought to bear towards chronic disease prevention, but rather the angle that I myself have taken in my academic research, and then subsequently trying to translate that into the real world with Myota, which I'll touch upon briefly, but I'm happy to discuss in the Q&A section as much as needed. So just to give a little bit of context here, because what we're talking about is chronic disease at the end of the day in terms of a pain point, and I think you're all familiar with the concept. Just a few high-level stats then to, you know, increase the sense of urgency if it isn't there already. 74% of all deaths globally are associated with chronic diseases. And if you take all adults on the planet, 1 in 3 of them suffer from at least one of these diseases. To take a very specific case of a chronic disease like type 2 diabetes, the National Health Service in the UK, the NHS, spends £1 million an hour on type diabetes, and this is a growing problem throughout the world. So we are, you know, rapidly running out of options in terms of how to deal with this issue. I think you've probably heard in many places that this is bankrupting our healthcare systems. 95% of US healthcare spend is on chronic disease. So I think you know, we're sorely needing some solutions here. And one of the things that's happened in the past, well, it's hard to give you an exact number, but 25 years, is that DNA sequencing technology has improved, which really allows us to explore the microbiome in particular and how that can play into disease like never before. So on the left here, you've got a commonly shown figure, which is essentially showing the price of sequencing a human genome through time, and Moore's law, which was for you know, computing chips, was devised for computing chips, is shown here as a reference. You can see that we've dropped the cost of sequencing at a rate that's faster than Moore's law. This is what has actually allowed us to go from $100 million genome to one that costs less than $1,000 today in less than 2 decades. And what that means is that all of a sudden we can sequence the DNA in lots of different environments, including different parts of the human body. And the result is on the right, an explosion in the number of publications exploring the human microbiome, and specifically the gut microbiome, because now we can actually sequence it by taking a swab of any body site or body fluid, and then sequencing the DNA in it, find out exactly what kind of bacteria live there, which is the first step to getting a descriptive map of how the microbiome works and impacts health. But, you know, for those of you who aren't super familiar with the gut microbiome is actually really important for our health. Inside our guts, and specifically our lower intestine, our large intestine, and the colon in particular, live a huge amount of bacteria, about 1 kilo of them at any one time. And I mean, if you go through the different compartments of our gut, you can see the increasing concentration or numbers of bacteria as you move from the stomach, where you have about between 10 and 1,000 bacteria per milliliter. You're getting to 1 trillion by the time you get to the colon. And that's where the vast majority of our gut microbiome lives. What is the gut microbiome? It's actually more than just bacteria. It's essentially all microorganisms that lives in our gut, and consists of bacteria, which are the most perhaps interesting from a medical perspective, but there are also fungi and archaea, which are a different type of microorganism. And these together form the human gut microbiome. We're talking about as many cells, as many bacterial cells in particular, as there are human cells in the human body. Of course, bacterial cells are much smaller, as I'm sure you all know, but this is quite a lot of stuff in our guts. That's actually, if you really want to get cheeky, it makes you ask how much of us is actually human, because 200 times the genetic potential of our genome exists in our gut through the genomes of our bacteria that live there. And so this, in fact, is the first point that I'd like to make about the microbiome in terms of its ability to modulate human health, is that it has, just loosely speaking, about 200 times the gene content of our genome, the microbiome that is, and therefore 200 times or more the chemical and metabolic potential and capability to do things with its environment and with dietary compounds, which is the main thing that it interacts with. So really, this is an important organ that we're now appreciating as such, and that has many roles to play in our health and disease. Just in terms of very high-level factors that influence the composition of the gut microbiome, if you weren't aware of this already, obviously your diet is probably the #1 variable that impacts it on a daily basis, since if you view the gut as a tube and bacteria living in it, they're exposed to our diet on a daily basis and feed on its remains, in particular dietary fiber, which I'll get into in a second. But another couple of things that really have a lot of impact are birth conditions. So you may have heard that when someone is born by cesarean section, then they aren't inoculated with the same microbiome as someone who's born in a normal traditional delivery. They're rather inoculated with microbiome that resembles their mother's skin. And that has actually been shown to have impacts in terms of predisposition to allergies and respiratory infections in the first year of life. So even the moment of birth is a very, very important one for the microbiome, and one that we're only learning now how to deal with. And the last thing, you might be very well aware of this, but antibiotics, they're unbelievable in terms of what they do in terms of damage. We take antibiotics very easily these days, and I'm not saying we shouldn't. They've saved countless lives. However, when you take oral antibiotics, you're essentially taking a nuclear bomb, especially when they're broad spectrum, to your gut microbiome, and it's going to kill bacteria indiscriminately, good ones and bad ones, which therefore means that after you've finished antibiotics, your microbiome will look very different and will repopulate in a new way. And that can have all sorts of, if you believe, an association between the microbiome and disease or your health and other parameters, then having a new microbiome right after antibiotics means, surely it means something. And that's something that, and indeed that's being shown. And I think that's just food for thought, that there's one thing that you remember from this as a piece of advice, is that next time you take antibiotics, make sure to eat a lot of fiber afterwards. Fiber is the main food for healthy gut bacteria, and it'll help them get back in place. But so fiber, this is one of the core functions of the human gut microbiome, and where I want to focus most of our discussion today is actually it does many things, but why do we allow it to live there, and why does it live there? Why do these bacteria live in our guts? They live there because they're opportunistic. They're living there because when we eat our food, we can't actually break down dietary fiber. It travels through our guts all the way to our colon, and there the bacteria are waiting for it because they actually can. I mentioned they have a lot more chemical potential than we do. They can break down dietary fiber. They can ferment them into what are called short-chain fatty acids. The vast majority of fiber biomass gets turned into short-chain fatty acids in the colon, and these short-chain fatty acids have lots of impact on our body. In particular, they are the dominant energy source for colon cells, which is pretty amazing. I mean, perhaps many of you knew this, but I think it's an incredible fact that the main energy source for the cells of our colon, of our large intestine, comes from a byproduct of the fermentation of fiber by the bacteria that live there. We can't feed our colon cells without feeding the bacteria that live in our guts with fiber. And so this already should be a reason to eat lots of fiber, because otherwise these bacteria start turning on the gut lining itself and start chewing at it. So yeah, it's good to keep them happy and give them lots of dietary fiber. But other roles of short chain fatty acids is that they, as I say, they strengthen these junctions between the cells in the gut lining, and loose junctions have been associated with a condition that people sometimes call leaky gut. It's considered a dubious term in some medical communities, but certainly there is such a thing as leakiness of the gut membrane, which if that happens can lead to increased inflammation, susceptibility to infection, and all sorts of other conditions. So strengthening that gut lining is very important, and that's one of the key roles of these short chain fatty acids. But when it comes to chronic disease prevention, well, they do two things that are very important. One, short-chain fatty acids are anti-inflammatory. They're highly anti-inflammatory. When you have short-chain fatty acids produced, they will exert anti-inflammatory pressure on your immune system and maintain low levels of chronic inflammation. And chronic inflammation, as you may know, is associated with all sorts of conditions, chronic diseases, including inflammatory bowel disease, depression, anxiety, neurodegeneration, even diabetes. And then on the other hand, short-chain fatty acids are actually impacting glucose metabolism as well. Totally different mechanism, but they're also doing that. And glucose metabolism is, of course, really important to keep healthy in order to prevent things like type 2 diabetes and other metabolic conditions. So that is actually two distinct mechanisms by which these short-chain fatty acids impact health. The last one that's worth noting is that they keep the pH in the colon relatively low, which is favorable for mineral absorption, iron absorption in the gut, and so on. So that all sounds like a lot. And I think one of the things that I would ask, or that I have been asked and that I think is a good question, is how is this possible? This sounds like it's too good to be true. These short-chain fatty acids are doing so much. Like, how could this be? And I think the only answer that I have to that, which I believe is the answer, is that this is the product of millions of years of evolution between our bodies and the bacteria that are allowed to live in our guts. They have opportunistically camped out there to feed on the fiber that we eat, but we let them stay there because then they make these goodies, short-chain fatty acids, which our bodies have evolved to use in a variety of different ways, and that ultimately form the basis of our symbiotic relationship. It's a pretty crazy kind of framework, but this is the world we live in. And if it sounds very strange, all you have to do is think of cows or other ruminants who get all their calories from bacteria breaking down fiber that they can't break down themselves. So this is, not a foreign concept, let's say, to the animal kingdom in general. At any rate, how do we study this process? How do we better understand this? Well, that's what I got into as a postdoc, and what led me down this track is, okay, we've got short chain fatty acids produced in the gut. That's super important. Can we study this process? You can't study that with DNA sequencing. You know the bacteria do this. So instead do this as a function, but you have to study this in the lab. So what we do is that we collected lots of stool samples. That's the way that you study the human gut microbiome. You get stool samples that are freshly passed, so the bacteria is still alive. And just for reference, by the way, stool is about 50% bacteria. So it's really a lot of bacteria you're getting there that represent a snapshot of your gut microbiome. And we essentially homogenize that and played out the slurry into plates like these, into which we can spike in different fibers into different wells and study through time how many short chain fatty acids are produced in each well. Which bacteria are breaking down the fibers is hard to tell because they're all in there at once. But what we can still find out is which fibers are being broken down into short chain fatty acids and at what rate. And we can study many different fibers in parallel in a single person. So one individual's microbiome can be queried in this manner in order to quantify the fermentation capability, if you like, which is essentially its ability to break down different fibers into short chain fatty acids, which have all these disease preventing and health promoting properties. In principle, what this does is that it allows us to identify which fibers are optimal for an individual as well. So that's that is an opportunity for clinical impact there. But so what do we find if we do this? This is a snapshot from some work that we did at the company Myota that we built based on this, where we designed precision blends of dietary fibers to treat and prevent chronic disease. We studied many different fibers. Each fiber is shown here by some acronym. And on the y-axis, you've got the production rate of butyrate, which is a short chain fatty acid. In fact, it's, people's favorite short chain fatty acid. It's the one that's most anti-inflammatory, the one that has the most potential for human health, and is fundamental in its generation. You can see here that you've got the averages over about 80 people here for a given fiber shown, and the production rate of butyrate from that fiber with standard deviations shown as error bars. So the differences between individuals is quite significant. And this is, I think, a very important point. We all have different bacteria. We all have different microbiomes. And so we have different abilities to break down the same fibers, which in principle means that either you need to account for this difference between people, and when you create blends of different dietary fibers to promote short chain fatty acid production, and that's what we do at Myota, or you have to personalize it based on the individual's microbiome. That's technically feasible, but operationally very difficult, because all of a sudden you go from something that's really cheap and effective as a solution, namely dietary fiber, safe and accessible, to something that becomes complex operationally, designing blends of fibers on a personalized basis based on diagnostics. And that inflates the price, it complicates everything. But these are the data that I think are really relevant to us understanding, leveraging this process towards disease prevention. A couple of other things that we find out about fibers when we look into detail is that the different chain lengths of the same fibers, what people call the same fiber, actually affect its ability to be fermented into short chain fatty acids by the gut lining. So for example, you've got these two here, inulin and phos. Both of those are polymers of fructose, fructans essentially. And the difference between inulin and phos is merely one of chain length, the number of fructose monomers inside that fiber, but otherwise they could just be called fructans together. The fermentation profile, though, very different. This is propionate, a short chain fatty acid, and the production rate of propionate on the y-axis, and these are different fibers. For a given individual here, only one person. See inulin, that person makes no propionate at all from inulin, but lots from phos. So a shorter chain length makes it accessible to the microbiome to ferment into propionate. Similarly, this chitin fiber, chitosan, which is a particular fiber that you can find in chitin, and microchitosan, which is essentially a broken down version of it, are in the latter case highly fermentable, and in the former not. So the point is, we can't just eat any old fiber and expect short chain fatty acids to come out of it. It's actually rather specific to both your microbiome and what the specific fiber type is. So this is something that we have to study with a certain degree of precision in order to design strategies that are going to be effective in some way. So here's a very specific example of how the fermentation capability of an individual might have some impact in the context of chronic disease. These are very preliminary data. I want to emphasize that because this is done in the context of a collaboration. We haven't published these data yet, and they're, like I say, very preliminary, but definitely in the direction of the science that we were talking about earlier, and show a link with potentially one of the more frightening chronic diseases we have, namely Alzheimer's disease, which is not really in the forefront of people's mind when they think of chronic disease, but actually one that is, nor gut health, but is actually related, potentially in a very significant way. So just a bit of background about Alzheimer's. I mean, I don't need to introduce Alzheimer's disease to all of you. I'm sure you're all familiar with this. But one thing that surfaced in the past years in Alzheimer's research is that we've known for a long time there are these plaques, amyloid plaques, that are deposed in the brains of patients. And you can see these on brain scans and in autopsies. However, those, while we used to think were linked to the disease etiology very closely, are not that well correlated with cognitive decline symptoms. Rather, what has been shown is that levels of neuroinflammation, so inflammation in the brain, is most strongly correlated with cognitive decline symptoms, and less so the plaques. However, it's worth noting that, as I mentioned, short-chain fatty acids are anti-inflammatory and they circulate in the blood. They also have been found in the cerebrospinal fluid. We find that actually levels of acetate and valerate, both of which are short-chain fatty acids, in the blood are positively associated with the levels of amyloid deposition and neuroinflammation. And butyrate, the anti-inflammatory short-chain fatty acids, as I mentioned, is negatively associated with that. Namely, the more butyrate you have in your blood, the less of these plaques you have, and also, as it was shown in a separate analysis, less neuroinflammation and cognitive decline symptoms you have. So that's all just to say the levels of short-chain fatty acids in your blood are correlated with those things. No causal link, but that's already interesting and makes a certain degree of sense. So what we did is that we tested the fermentation capabilities of 12 volunteers from a particular cohort of elderly individuals with early onset or established Alzheimer's disease and a particular mutant status on the ApoE gene. ApoE gene is actually a gene that's the strongest predictor of Alzheimer's disease genetically. If you have a particular variant of this mutation, you're highly likely to suffer from early onset Alzheimer's in your life. And so we looked at 12 volunteers that had this ApoE mutation status. And what we found is as follows. But before I get into the details, I just want to explain how these data work. First of all, we have these plots where you've got on the x-axis different fibers, individual fibers, so inulin, pectin, phos, and so on. It doesn't matter what the individual fibers are, but each column represents a fiber. And each row, an individual. The more red that square is, the more that fiber can be broken down into butyrate, for example, in that person. And we then standardize these scores into what are called Z-scores within that cohort. And then if we segment people by their mutation status, what do we find? We find no clear, so the top five rows patients suffer from this mutation that is more likely to lead to Alzheimer's disease. Nothing clear comes out of butyrate, but if you look at valerate, which as I mentioned earlier was associated with plaque deposition and so on, you can see that those who have the epsilon-4 mutation have higher rates of production of valerate across the different fibers than those with other mutations. Not enough, so more red in this cohort essentially than in these other people who don't have the mutation. Again, qualitative data, we don't have enough people, but these data are consistent with what we see in the blood of patients. So it seems that somehow the ability of your microbiome to produce short-chain fatty acids from fibers and which short-chain fatty acids from which fibers has an impact on your levels of short-chain fatty acids in your blood and body, and in this particular example, potentially a very significant impact on your tendency to contract Alzheimer's disease or get dementia of various types. I think that link is not at all obvious to anyone, including myself, exactly how the mechanism works. But this is just to show you an arc that shows the potential of this process in our guts that's happening all the time. And just another reminder to eat your fiber when you get a chance. But so I think that one of the points that I've made so far is that temporal dynamics and personalization are actually pretty important factors to consider in this. I mean, I think this is consistent with our understanding of the role of nutrition in general. Postprandial inflammation, for example, by postprandial, I mean following a meal. Inflammation following a meal is actually very difficult to study these days. And so we're trying to understand, how does food and dietary fiber and compounds like this impact your levels of inflammation as one endpoint. But that's very difficult to do because you just have a single time point. If you do it as in a traditional manner, people get a blood test before and then after a certain time. But we don't know which biomarkers to study, which inflammation markers respond to meals, how long it takes for them to respond. Is there a big difference between people? We just know that physiological responses to meals in different individuals are extremely personalized. They differ by people and they're dynamic through time. So for example, here's an example from glucose monitoring, checking how much glucose is present in the blood in a particular cohort of individuals in response to two different foods. So a banana and a cookie, banana in yellow. This individual here you see has a sugar spike in response to the banana, but not to the cookies. And vice versa, this individual has a sugar spike from the cookies, but not the banana. So personalized responses and dynamic in time, right? Similarly, if we study microbiome composition changes, which this is actually a snippet from a study that I'd conducted a while ago, if you feed people a particular fiber, you see a bloom in this particular Bacteroides uniformis. It's a particular bacterium in the gut. And this is through time, its abundance over a week-long intervention period. You see that after day 3, when people start getting given this inulin as a supplement, that bacterium blooms in the cohort that were given inulin. But it doesn't bloom in those who weren't given that fiber. So that seems to run counter to what I just said. Looks like it's working on average across everybody, except that if you break it down by individual, you can see some people have a small bloom, others have a very large bloom of that bacterium. And it depends on the person, on their immune system, and countless other variables. Even if you try and control everything, it's extremely hard to take an engineering approach with the human body because there are too many variables, too many things going on. So I just wanted to make the point that Personalization are really important. We talked about the personalized fermentation capability of an individual, their glucose responses, their microbiome changes, their everything is personalized. And so one of the things that we wanted to look at in more detail is, are inflammatory responses personalized as well? So here is where we did this particular study called HIPPIE, which is an acronym for High Resolution Investigation into Postprandial Inflammation Biomarkers. So HIPPIE is a bit of a stretch, but I'm sure you're all familiar with how these acronyms work. The idea was just to study 3 different individuals and their ability to, or rather their inflammation status in response to 3 different test meals. The first meal was a Mediterranean diet meal, essentially one which, if you're not familiar with that type of diet, well, I'm sure you are in the region of the world you're in, but what I mean is in the field of dietetics, the Mediterranean diet is considered the most anti-inflammatory and longevity promoting diet. And so we picked that one as an anti-inflammatory meal. Then we have the second meal, which is literally McDonald's, like junk food, as bad as we could make it. And the third one was that same junk food meal, but with 20 grams of dietary fiber supplement that was based on the person's ability to break down fiber and designed to boost butyrate, which is anti-inflammatory. So essentially a highly pro-inflammatory meal, a junk food meal, and then a junk food meal with fiber added back in to try and make it anti-inflammatory again. And we studied this, actually, each meal for a 28-hour period, they had a blood line in, and we were taking blood samples through time. So we didn't look at single snapshots, as I mentioned before, but rather continuous monitoring of those inflammation biomarkers, which was quite a lift. But so the endpoints that we looked at were 92 different inflammation biomarkers in the blood or plasma, as well as studying short chain fatty acids in the blood to see if they were in any way accumulating based on the meals that they consumed. So just at a very high level, to start, if I make this plot, just to explain this plot, essentially these are the 3 time series concatenated. So you've got 3 time series, 1 for each color. Red is the first meal, blue is the second meal, and green is the second. And a row is a particular biomarker. So there are there are 92 different biomarkers. Red is high, blue is low, and you can see it changing in time. Whereas each column is a time point, right? So you can kind of view that red chunk as the time series for a given biomarker here on each row. And I mean, here, it's not to look in great detail, but rather if you squint a little, what you'll see is that this is for participant 1, this is for participant 2, participant 3. And so again, I'm going to go back and forth for a second. If you go participant 2, you've got quite a lot of blue in response to the meal A and quite a lot of red in response to meal B. And that's flipped in participant 3. So again, very qualitative. I'm not making any quantitative statements here, but you can clearly see that participant 2 has more of an anti-inflammatory response to meal A and more of a pro-inflammatory response to meal B, and vice versa in participant 3. So already at a very high level overview of all these different biomarkers of inflammation, you can see that people respond differently to exactly the same meals. And this is the point I was trying to make, but shown a little bit more clearly. So then we went and looked into different biomarkers and tried to categorize them by their response profile. We went through the list and then classified them into either inflammation biomarkers that were meal responsive or showing some response to the meals that the patients were consuming, or non-responsive. And then those that were responsive, they were classified into either personalized responses, where people had responses that were not necessarily the same across individuals, or responses that were common across everyone. And similarly, those that were non-responsive biomarkers were either static, namely they didn't change in time throughout the time series considered, or dynamic, but following some cycle that was unrelated to the experiment and rather more something like a circadian rhythm cycle, like your body clock or something like that. So some examples. Here's a static biomarker, this beta NGF. On each row is an individual. So the first top row is the first participant, second row, second participant, third row, third. And each column is a meal. So meal A on the left, meal B on the right, on the middle, and C on the right. And just if I remind you, meal A is the anti-inflammatory meal, meal B is the pro-inflammatory one, and meal C pro-inflammatory plus fiber. Here, no response in this biomarker, irrespective of the meal or the participant. But here's another one that has really nice responses, but it follows basically a 24-hour cycle. The red line is when the meal was consumed, and you can see essentially that this biomarker's value cycles from one level through the 24-hour period back to that same level. And so this is much more likely to be related to hormones, for example, than any experiment we were doing. Here's an example of a biomarker that was responsive, the same response to all individuals. A spike right after the meal, independent of the meal and independent of the person. As it turns out, it's a protease inhibitor that's secreted in our saliva. And so this makes a lot of sense. When you're eating something, it's going to be secreted. No clear reference to what you ate, just that they ate, and that's happens to be the case. But then when we get to the interesting stuff, here is where we come back to our original point, that things are personalized. So IL-6, you may already know this biomarker, is a very important pro-inflammatory cytokine. So it's a pro-inflammatory signal in your body. And you can see that the responses for participants 2 and 3 are essentially flat, except for this blip here, whereas participant 1 has a pro-inflammatory response after about 5 hours following the meal in this green window here, where you've got a spike in IL-6 concentration in their blood. And that goes away when you add the fiber. So again, N of 1, but nonetheless significant to note that only one of the 3 individuals had a pro-inflammatory response to the McDonald's meal, and that went away when you put fiber back in. Same thing happens with IL-8. IL-8 is also pro-inflammatory, but a very different kind of protein. It sounds like it's the same because it's got the same name almost, but they're very different. So there's no, I guess what I'm saying is this is measuring something entirely orthogonal and still seeing the same thing, pro-inflammatory response to meal B in the first participant. Same thing with MCP4, a different biomarker. Pro-inflammatory response to meal B in that first individual goes away in meal C. Something happens sooner, but that's I can't explain it. All I know is that the pro-inflammatory response 5-6 hours after the meal does go away. IL-10 is an opposite signal. It's an anti-inflammatory cytokine. And here we see the opposite effect. Participants 1 and 2 don't have any IL-10 or anti-inflammatory signal, either from the Mediterranean anti-inflammatory meal or the junk food meal. But participant 3 does, and actually has a really nice anti-inflammatory response to the Mediterranean diet meal. No such response to the junk food meal, though. And the key point is only the 3rd participant had this. So all this to say that clearly we're seeing some personalized responses in otherwise pretty bread and butter inflammation biomarkers in response to what's generally accepted is, anti-inflammatory food versus pro-inflammatory food. Something to keep in mind. But then what we also did is we analyzed short-chain fatty acids in the blood. And so short-chain fatty acids are these byproducts of fermentation of fiber, as I mentioned. They make their way into the blood. And one of the big questions was, do these things actually stick around in the blood, or do they get metabolized by the liver the first time they go around? In which case, the opportunity for impact to chronic diseases is much smaller and more limited to the gut. Here we can see that butyrate is actually more or less flat with a noisy baseline until the meal, and then there's a nice accumulation of butyrate for a period following the meal, which actually, while this is a noisy trajectory, is, I think, really important to highlight, because this shows that no, butyrate is not immediately metabolized by the liver. It can accumulate in the blood. The dynamics are complex. They change in time, but if butyrate is exerting any anti-inflammatory pressure on other tissues than just the gut, which we know it does, because if you inject butyrate into a rat, you see gene expression changes in their brain, and that's directly traceable to the impacts of butyrate. So butyrate is actually circulating in the blood and staying there for some amount of time, which means it can impact other tissues as well. And the impacts of anti-inflammatory activity coming from short-chain fatty acids, coming from the microbiome, from eating fiber, are possibly going to affect other tissues as well, like the brain. And we saw a link before with the Alzheimer's disease cohort. So I think that this basically shows, and then if you scale that to all three meals and all three participants, you can see a similar thing, noisy trajectories. But if you see meal A, participant 3 here, very clear accumulation of short-chain fatty acids here in that meal. And this is, I believe, showing all butyrate, but we studied all short-chain fatty acids. Similar here, you can see this person has an accumulation after this meal, which is particularly, actually enriched in fiber, but fermentable fiber into butyrate. You don't see that as much, neither with the Mediterranean diet meal or the junk food meal without the fiber. You have to add in the fiber to see this, right? Nice spike, and the fiber that was specifically fermented into short-chain fatty acids, which also, parenthetically, makes us wonder, hey, how come they didn't make lots of the fiber in the Mediterranean diet meal into short-chain fatty acids? Well, maybe because their microbiomes that person's microbiome isn't well suited to ferment those particular fibers. Remember, in this meal, we picked the fibers based on their microbiome, and there we see a pretty nice signal. So anyway, this is complicated stuff, and in terms of translating this into the clinic, it's not clear at all from here, like, oh yeah, just measure short-chain fatty acids 6 hours after a meal, and that's when you see a spike, if there is any. It's not that simple, unfortunately. But I'm just using these points to illustrate the fact that if you take a single blood test snapshot, you're missing all these dynamics. You might miss a spike that's gone away by then already. You might be studying the wrong inflammation biomarker, as we saw before, for that person. It's really quite difficult to find the signal, but you have to look at individuals and temporal dynamics as well. So what can we conclude from that particular study? Well, we observed personalized responses on short timescales and in levels of inflammation to the same meals. So what you eat impacts your levels of inflammation. That, I think, is well known, but here we're showing it for the first time with actual biomarkers that are relevant to disease. And pro-inflammatory responses to a given meal are consistent within a person. If you're showing a pro-inflammatory response in one biomarker to a particular meal, likely a bunch of other biomarkers are on the rise as well. We also show the dynamics of short-chain fatty acids in the blood are non-trivial. They're not just flat, so you do see accumulations in time, which means that when they're produced in the gut, they're going into the blood and they're accumulating to a certain extent, which means that they might be relevant endpoints to study in clinical studies and clinical trials, studying chronic disease and the impact of fiber on that. And I mean, ultimately, that we really need to account for these personalized responses and these time-dependent dynamics in nutritional intervention studies moving forward, particularly if we want to get to a place where we can better understand, the impact of the microbiome in modulating health and disease. So the last point that I'll leave you with is that's all well and good. And I showed you a few snapshots of some scientific stuff that we've done here and there. And overall, that's just a drop in the ocean in terms of what everyone's been working on in terms of the gut microbiome and how it impacts chronic disease, mainly through this short chain fatty acid production, as I mentioned. But where we really want to get to is fiber is so important for health that it should, in a sense, carry health claims like drugs do, and like very important things for health do. The way that you get those, though, is you have to do clinical trials and show this. And so this is just a point for your consideration. It's rather than, it's just an open point. The European Food Safety Authority regulate food-associated health claims. We have the opportunity, based on the science that I showed you and all the other background science in the field, to leverage fiber and the microbiome towards foods with health claims. Say a food, imagine a food bar that carries on it the wording, this reduces intestinal inflammation and IL-10 concentrations in your blood, for example, which to a doctor means a lot. It might not mean that much to the individual consumer, but if they have a condition where this is relevant, that means a lot. Imagine a food product that's like medicine, right? And that is the potential of fiber. In order to get that health claim, you need to go through several clinical trials. You're a pilot study, followed by a pivotal study, probably a dosing study as well. You apply to EFSA with a health claim in mind. They might approve a given wording and you iterate with them until you've got a wording that you like. But here's the key point. You have to spend a lot of money to do clinical trials, a lot. And why would you do them if at the end of the day, the health claim you get is on a food over which you have no intellectual property, you have no exclusive rights over the health claim because it's tied to the food. So if you have an oats-based product and oats are shown to reduce a certain biomarker or some health claim, anyone who sells oats can use it. So we have a market failure, essentially. We have a problem in the market because there isn't the right incentive structure to create companies that will sell fiber-based food products that carry health claims, because no one's going to invest in the clinical trials to get these health claims, given that they can't own them afterwards. And this, I think, is one of the problems we face. The science is increasingly clear, but we have difficulty in translating this into the real world because you need a business model that is somehow protectable, has some kind of IP, and otherwise people won't invest in that business to grow it and actually lead it to a place where it can have genuine impact on public health. So with that, I'd just like to leave you with a couple of very high-level points. The gut microbiome, as I was saying, combined with diet is a really key modulator of our health. And I showed you a couple of details in the short-chain fatty acid space on how these might be able to impact us. And of course, nutritional solutions, leveraging this process, are really important preventative tools for managing this chronic disease problem we have. But in order that we get there, we actually have to innovate on the business model front and probably on the regulatory front in order to open this possibility to the widest set. I'd like to just very quickly acknowledge some collaborators, particularly on the HIPPIE study that was funded by the C-RAVE Foundation, and some collaborators at the University of Geneva, at Myota, the company that I was just talking about that we spun out to address this problem, and my postdoctoral advisor, Professor Eric Ahn. With that, I'd happily take any questions, thanks again for this great opportunity. It's been a real pleasure talking to you all.